In this lecture, I will talk about T follicular helper cell, how these cells are generated and what are the effector functions. So I will first talk about the T follicular helper cell, how these follicular cells actually generate. In the tissue, there may be infections and also pathogen can be present in the blood, direct into the blood. So these are the main primary route. In the tissue, there is already some sentinels like dendritic cells are present. If there is infections, the dendritic cell can sense the pathogen. These dendritic cells actually leave the infected tissue site to the nearest lymph node. So this is the route if there is infection in the tissue. So the dendritic cell engulf the pathogen and some dendritic cell leave the infected tissue site. This is infected tissue site. What happens? Here the dendritic cell actually activate naive T cells. So this is naive T cells. So this cell is inactive, and when this inactive cell interact with this dendritic cell, it becomes activated, and these activated cells are called effector T helper cell. This effector T helper cell could be TH1, TH2, TH17, and there is another route, that is blood. If the pathogen is present directly into the blood, what happens? From the blood, the pathogen reach to the spleen. Here in this case, it is lymph node or lymph gland. But in case of blood, the pathogen reach to the spleen, because blood-borne pathogen are actually trapped in the spleen. And in the spleen, there is already dendritic cells are present, and these dendritic cells perform the same function. They actually capture the pathogen. So if there is any in the spleen there is any infection, this dendritic cell actually capture the pathogen and represent to the naive T helper cell. In the spleen also, naive T cell actually recirculate. Actually they patrol through this spleen and lymph node. If they recognize any dendritic cells that is carrying pathogen, actually it is dendritic cell showing the pathogen fragment, actually antigen fragment by MHC2 molecules, by which the naive T helper cell recognize and it becomes activated. So from the inactivated form to activated form, it can be TH1, TH2, TH17. So this dendritic cell and the T cell interaction occur in case of lymph node. This area is called paracortex. But in case of spleen, this dendritic cell and naive T cells interaction occur in a region called PALS. The full form of this PALS is periarteriolar lymphoid sheath. Periarteriolar lymphoid sheath. When these cells become activated, this naive cell become activated, a very important thing occurs, that is these activated cells somehow come out from this paracortex and PLS to a region that is very close to another region that is called, this is actually follicle. So in the lymph node there is a follicle, and in the spleen there is also a follicle, and this follicle is very close, in case of lymph node and spleen, very close to this paracortex and PLS. In this follicle there is a cell present that is called B cell. B cells are present in the follicle. Okay, and what happens when this dendritic cell and naive cells interact? Some B cells also come out from the follicle and join these activated T effector cells. So B cell actually come out. This B cell come out close to these effector cells, activated effector cells. And now another thing occurs. So you can see, for the development of T follicular helper cell, first thing occurs, that is T cell activations. Another thing occurs, that is B cell activation. So these two cells interact and B cell get activated. There is two things occur when these effector cells and B cells interact. One is this B cell become short-lived plasma cell, and they actually class switch their antibody. I have already discussed about class switch. So this B cell actually class switch their antibody. At the beginning, these naive cells in the follicle, naive B cells are present. That means inactive B cells are present. But when these B cells get activated, it actually switch their antibody. At first, in the naive B cells, there was IgM and IgD antibody. But now this B cell can switch into different antibody like IgA, IgG and IgE. This is called class switching, but this class switching is not very extensive. And another thing that do not occur during this interaction, that is called somatic hypermutations. And for this somatic hypermutation, one thing is occurred, that is called affinity maturation. This is not occurred during these interactions. During these interactions, dendritic cell and naive T cells, some naive T cells interact with MHC2 molecule with strong interaction. Very important. I am mentioning it in red color. This is very important for T follicular helper cells. So I am talking about T follicular helper cell. What happens during this dendritic cell and naive T cell interactions? Some T helper cells interact with MHC2 molecule very high affinity, or you can say strength of interaction, high strength interactions. So what happens? During these interactions, these naive cells become activated effector cells TH1, TH2, TH17, but few naive cells interact with class 2 MHC molecule with very high strength. So some T helper cells interact with MHC2 molecule with very high affinity or strength interactions. Those cells that interact with this MHC2 molecule with high affinity will become T follicular helper cell. In this interaction, some cells become T follicular helper cells, TFH. Some cells become T follicular helper cells. These cells actually enter into the follicle. Okay, and what happens here? The short-lived plasma cells actually live from the spleen and lymph node, but few short-lived plasma cells enter into the follicle. So what happens? These short-lived cells, plasma cells enter. So B cell enter. B cell means activated B cells. This is activated. Activated small-lived plasma cells enter and T follicular helper cells enter. Now, in the beginning, this follicle is called primary follicle. But when this B cell and T follicular cell enter into the follicle, primary follicle become change into secondary follicle. And the secondary follicle is known as germinal center. And three important things occurs in the germinal center with the help of this T follicular helper cell. I am telling you, three things occurs. If these two cells interact with each other, then three things occur. That is, this B cell become long-lived plasma cells. Then the third thing is class switch occur, but now it is extensive. So here I make a tick mark. There is negative mark, cross mark. No extensive class switching occur. Class switching occur, but not extensively. Here extensive class switching occur. Another thing occur that is very important. That is somatic hypermutations. And for that reason, affinity maturation occur. Affinity maturations. And after affinity maturations, what happens? The antibody that is producing from these long-lived plasma cells can bind with the antigen, same antigen, but with high affinity. That is called affinity maturations. Okay, so this is the effector function of T follicular helper cells. Now I will tell you how T follicular helper cells are generated. The first thing is that they interact with MHC2 molecule with high affinity. Another thing, some interleukin are actually responsible, like IL-6 and IL-21. This molecular chemical, this interleukin, actually helps in the production of T follicular helper cell, because these two cytokine actually helps to induce a master regulator in this T follicular helper cell, that is called BCL6. This BCL6 master regulator, that is actually transcription factor, can inhibit T-beta transcription factor, can inhibit Rod gamma T, can inhibit FOXP3, can inhibit all these transcription factor. It can inhibit GATA3. So it can inhibit TH1, TH17, TREG. It can inhibit TH2. So this molecule can inhibit all other cell type. Okay, and this T follicular cells can also secrete cytokine, that is IL-4 and IL-21. And the main function of this follicular helper cell to produce this primary follicle into secondary follicle or germinal center and activate the activated B cells another time, that this T follicular helper cell actually express a molecule called CD40L, and when this CD40L interact with this B cells, a molecule in the B cell called CD40, when these two molecules interact, CD40L from the T helper cell and B cell produce CD40. When these two molecules interact, these two things occur, that is called class 2 switching, that is extensive and somatic hypermutations. Another thing is, in this B cell activations, this CD40L and CD40 interaction also occur. This T helper cell, that is effector helper cells, also attack these B cells with this CD40L and CD40. So there is also a interaction occur between the CD40L and CD40. But T follicular helper cells actually attach with the CD40 another time with the CD40L. One important phenomena actually occurred if there is any mutation in the CD40L. If there is any mutation in the CD40L, one thing occur, that is called hyper IgM syndrome. Now you can understand why it is hyper IgM syndrome. You can see T follicular helper cell actually activate the B cell to produce class switching, so that it can produce IgG, IgA, IgE antibody, different type of antibody. Class switching occurs very extensively. But if there is any problem in the CD40L, so this interaction will not occur between this T follicular helper and B cells. But if there is any problem in this molecule CD40L, this B cell do not go through class switching, so it can only produce IgM. So hyper IgM in serum, hyper IgM is actually observed, not any other antibody type like IgA, IgG and IgE. So this is called hyper IgM syndrome if there is any mutation in the CD40L.